Hello everyone, my name is Lorena Derezanin and the topic of my talk today is the genome assembly of the tropical mustelid thyra and its adaptive genetic variation. So first, a bit of a background story. I'm sure you're familiar with carnivorans. This is a mammalian order adapted to various habitats. Its members occur on all of the continents and also utilize different ecological niches, from aquatic piscivores such as otter, to a border of frugivores such as kinkajou. Among carnivorans, there's this family called mustelids, which is the largest and most species-rich family that went through adaptive radiation. And the topic of the talk today is the Golonina. It's a subfamily of Mustelida, and it's our model group to study adaptive traits. So our aims and objectives were to assemble a high-quality reference genome for Thyra, and to compare it with two other Golonine species for which genomes were already publicly available. We specifically looked at positive selection on single genes, gene family expansions, and also larger structural variants. So when it comes to our Golonine species of choice, so beside Thyra, we took Wolverine for a comparison as a representative of subarctic habitat, and sable as an intermediate habitat representative. And beside certain traits that are common among these and shared among these species, there are certain species-specific traits that we're going to focus on today. For example, in Thyra is a non-seasonal breeder. It goes through multiple estresses throughout the year. Also, as a tropical inhabitant, it lost its adaptation for embryonic diapause, which is evident in other Golonine species and also in most of the other mustelids. And also there's this phenomenon known in Thyra. It's caching of unripe fruit. So Thyra caches a fruit that it's not ready to eat and then comes back and consumes it. When it comes to Wolverine, its ability to store large fat deposits is quite remarkable, and also it is known to form a response to nutrient-poor environment that it inhabits in the form of bone mass and density regulation and also macroautophagy. And sable is among mustelids known to have exceptional hearing, which it uses beside of olfaction for hunting prey in snow, under snow cover. And also what sable is mostly hunted for is its very fine, almost silky fur, and also diverse coat coloration among populations of the species. So just briefly about a bit about the phylogeny of these species. So Golonina originated in Eurasia and then rapidly spread across the continents. And today we're seeing Thyra as a basal Golonine species among the extant species. Here you can see in the distribution map of the species. So Wolverine has a large area, but the species, actually the individual that we used for our comparison, comes from this western part of Sweden. When it comes to sable, we use for the comparison individual coming from the northeastern China. And also we, for the structural variation analysis, we relied on the reference genome sequence of domestic ferret, which is equidistant from these species. A bit about the genome assembly. So we use 10x genomics linked reads, which are essentially barcoded Illumina short reads, which are mimicking long read technology. We assemble it with Supernova assembler. And when you check some of the metrics, I would say the most important here is the scaffold N50. So we have a lot of large scaffolds. Actually, the whole genome size of 2.4 gigabases. It's contained in 170 scaffolds, and also the haplotype block that could have been phased. It's quite large, so this is already quite solid for this genome assembly. Here in the upper pane, what you're seeing is the gene completeness assessment that we did to see how complete our genome is in terms of conserved genes that could be found among the species. So the Thyra actually is, in terms of fragmentation, quite good compared, for example, to Wolverine, which has a higher fragmentation of the genome and also a higher level of missing conserved orthologs. Here on the lower pane, you're seeing this synteny representation between domestic ferret genome and the Thyra. And even though here you can see some of the scaffolds, most likely in the Thyra genome, are being inverted, but overall we have 95% identity with domestic ferret genome. So a bit about the results. I'm not going to be able to go through the details of the methods that we used, but as I mentioned, we looked at positive selection in single genes. We looked at gene family expansion and also structural variation that we're going to talk a bit about more on the next slide. So among a lot of genes that we found, that we expected to be found in these species to be found under adaptive selection, are immune system related genes and olfactory genes. There were, of course, lower detection in Wolverine genome due to its fragmented genome. But overall, the interesting and most significant results are actually related to genes associated with those species-specific traits that I mentioned. So, for example, in Thyra, we found a certain number of genes related to reproduction, and more specifically, formation of placenta and embryo implementation. For Wolverine, we found a large number of genes related to actually associated with diet and energy conversion, and also muscle function. And a lot of them had to do with formation of adipose tissue and storage. And also, as I mentioned earlier, this nutrient deprivation response to a nutrient-poor habitat that Wolverine is inhabiting is also found to be in a form of bone density and mass regulation in Wolverine. When it comes to sable, we found a certain number of genes related to hair follicle development, which most likely has to do with this fine structure of sable fur. And also we found a certain number of genes associated with hearing, which is exceptional apparently in sable. So when it comes to distribution of structural variants in Thyra, here I just wanted to show you the extent of these homozygous structural variants affecting protein coding genes in Thyra, which you're seeing here. These are structural variants larger than 50 base pairs. So these are deletions, duplications, and inversions. What you're seeing here on the y-axis is reference domestic ferret chromosomes, on which we plotted these variants that we found in Thyra genome. And what we're suspecting here, actually these homozygous structural variants are usual suspects when it comes to local adaptation and speciation, and we're actually concluding that they're most likely drivers also for local adaptation in case of our model species. Just a couple of more notable findings for a concluding slide. In Thyra, we specifically found the duplication of the gene here that you can see. This gene is coding for the conversion of arsenic to less toxic compounds, and this came as a surprise to me when I first found it in Thyra. But then after literature research, I've noticed that high concentration of arsenic are actually not a new thing for Central and Latin America, which is the habitat which is a home range of the Thyra. So arsenic coming from this geological sources is known to affect human population in this area, and this is potentially something that Intyra developed as a response to this highly toxic compound in its environment. When it comes to another thing that is interesting in Tyra, there has been this paper from 80s cited for quite a long time, suspecting that Tyra has poor eyesight, which I think it might not be true. We found various vision-related genes, and especially one that appears to be rapidly evolving, and it has to do with a lens formation. Also, additionally, as I mentioned, this phenomenon of caching of unripe fruit in Tyra. So beside vision and olfaction, probably Tyra also relies on the memory and learning of the sites where it actually cached its unripe fruit. This is extensively discussed in this paper here. What is remarkable is that Tyra actually somehow manages to distinguish between different stages of the maturity of the fruit, and also different kinds of fruit in different stages. And it caches it and then comes back to consume it when it's ripe. So this is a potential indication of prospective thinking, but I guess some additional assessments need to be done. So this is all for me today. Thank you for listening. I will be ready for your questions, and also many thanks to my supervisors and collaborators who were putting a lot of effort and worked on this study. Thank you and bye.